at 9 a.m. PST. It's now, and I have the pleasure to welcome everyone to another Global IMMUNOTALK. We are back after a brief break, re-energized, and most importantly, with an amazing list of global immuno speakers that will be presenting in the next few months. Just as a reminder, Gita Stockinger will be presenting next week, so I hope you can all join us. So I will now stop sharing the screen because I would like to introduce to you our fabulous speaker today, Dr. Chen Dong. So Chen is originally from Wuhan, China, and is currently a professor at the Institute for Immunology at the Tsinghua University, and also more recently at the Shanghai Jiatong University School of Medicine. So Chen has served as a professor of immunology and the director of the Center for Inflammation and Cancer at the University of Texas MD Anderson Cancer Center before he moved to China, and also was for 4 years, from 2016 to 2020, the dean of the Tsinghua University School of Medicine. Chen research, as many of you may know, focuses on understanding the molecular mechanisms whereby immune and inflammatory responses are normally regulated, and to apply this knowledge to understanding and treatment of infection, autoimmunity, and allergy disorders, as well as cancer. And we can say without any hesitation that Chen and his team has made tremendous contributions in immune regulation, particularly on the field of T-cell differentiation from both his laboratories at US and China. As an example of Chen's impactful contribution to this field, we can highlight his work that led to the discoveries of Th17 cells and T follicular helper cell subsets within the immune system, and the elucidation of these subsets' biological and pathological functions. So Chen has over 200 publications in top-tier journals and was rated highly cited researchers for 7 years, from 2014 to 2020. The honors he has received include, in 2009, the American Association of Immunologists BB Bioscience Investigator Award, and in 2019, the International Cytokine and Interferon Society BioLegend William Paul Award. He's a member of the Chinese Academy of Sciences and a fellow of the American Association for the Advancement of Science and the Chinese Academy of Medicine. He's currently an editor for Immunity and a scientific advisory board member for MED, an editor-in-chief for Frontiers in Immunology, and an associate editor for China Sciences Life Sciences. On a more personal note, I have great memories when I had the pleasure to meet Chen at the Gordon conference. He and Carola Vinuesa invited me to attend, was particularly on T follicular helper cells, and I could experience their Chen's amicable and generous personality. So Chen, it's a great honor to have you as a global immuno speaker today, and we are really thankful and appreciative of all the efforts you put in preparing this talk. And we cannot wait to hear it. Chen's talk is entitled T-cell regulation in immunity and diseases. So, Chen, before we let you share the screen, your slides, as you know, we have a question that we would like to ask you. So we get to know you better and know your perspective. particularly here on how do scientists work together in the COVID-19 pandemic. What are your thoughts about that?
First, Nina, thank you so much for inviting me to this global IMMUNOTALKs. It's a wonderland for immunologists to get reconnected again. I think the question you asked is a very important one. So we first need to stay connected because we all miss each other. We shouldn't forget about each other. We saw names in journals, but we need to see each other in faces, virtually or in reality. And that's first thing. I think we need to stay connected. And the second thing is we need to work together because we all experience the globalization of sciences, but now everything is interrupted by COVID. I think we need to think of ways to resume our scientific collaboration, scientific exchange, and I think that's important. And I'm sure that scientists still need to work together in not only dealing with the infection, but also many challenges around the world. So I think we need to reverse the deglobalization of sciences and stay connected. Thank you.
Yes, thank you. Thank you so much for sharing those thoughts. And I couldn't agree more how, you know, science is a global endeavor. And yeah, borders are non-existent in science. And that's a beautiful aspect of it. So thank you so much for sharing that. Okay, Chen, we are all ready for your talk. So if you want to share your screen.
Yeah.
Perfect. And on a presenter mode. Okay. Excellent. Perfect. Thank you so much.
Okay, thank you. Good morning, everyone, wherever you are. In China, it's midnight, so it's morning again. So I'm sure most people are still in the morning. So and except European people. So my name is Chen Dong. It's my honor to present in this global IMMUNOTALKs. I found this wonderland, and I'm sure I will tell others to join and to listen. And as I said, stay connected. So today I will share with you two stories we recently published and on IL-17 producing T cells and also related lymphocytes. So Th17 cells were added to the map of CD4 helper T cells in 2005, more than 15 years ago. We and others proposed this represent the third lineage of helper T cell differentiation. These cells are named because they secrete IL-17, IL-17F. Also, they express a discrete array of cell surface receptors. Also, the differentiation of Th17 cells occurs independent of that for Th1 or Th2 cells. We know that Alina's favorite cytokine IL-6 together with TGF-beta would induce early Th17 cell differentiation, and we reported IL-1 also acted early to sustain and to mediate Th17 cell differentiation. Downstream of IL-6, of course, STAR3 is necessary, and also there are at least two Th17 specific transcription factors. The major one is ROR gamma T, discovered by Dan Quam and Dan Littman to be necessary and sufficient for Th17 cell differentiation. The other one was discovered in our lab, our alpha, which plays redundant and also coordinative roles as our gamma during Th17 cell differentiation. So initially, when we proposed Th17 cells as a distinct lineage of helper T cell differentiation, we also propose that these cells may have different biological functions compared to those by Th1 and Th2 cells. So in summary, Th17 cells are very important for our host defense to infection. We now have patients who have deficiency either in producing Th17 cells or could not respond to IL-17. These patients are sensitive to various infections, particularly by extracellular bacteria and also by fungi. Th17 cells are frequently found at the mucosal surface. Not only they are important for mucosal immunity to infection, but also they help to maintain the mucosal barrier by stimulating epithelium cells to repair and also form tight junction. Most importantly, in the field is the relevance or the function of Th17 cells in chronic autoimmune diseases. Now targeting Th17 cells has been already therapeutically approved for the treatment of psoriasis and other autoimmune diseases in humans, proving that indeed these cells are very important in human diseases. So the work in the field recently has been focused on the pathogenic regulation of pathogenic function of Th17 cells. As I mentioned, Th17 cells can be found at the resting state in the mucosal tissues, intestine, for example, but they don't elicit strong inflammatory reactions in those tissues. On the other hand, in disease models like EAE, Th17 cells can migrate into central nervous system and to induce chemokine production and induce tissue inflammation. So it's not clear why Th17 cells sometimes are quiescent and sometimes and even tolerant to the environment, but why in autoimmune diseases they play very important roles in sustaining or initiating tissue inflammation. So several years ago, we began to investigate this issue by looking at environmental factor that we all are familiar with. That's fever, because I'm sure many sitting in the audience have all already been enjoying being tested for the body temperature. Upon infection, our body temperature would increase, and these will limit the pathogen propagation, and also has been linked with regulation of innate immunity, etc. But whether or not fever can directly regulate T cell responses was not very clear. So several years ago, the former postdoc, now associate investigator, research associate professor Xiaohu Wang in the lab decided to investigate whether or not high temperature could influence T cell differentiation. So Xiaohu incubated T cells at 37 degree or 39.5 degree to drive these cells to naive CD4 T cells into Th1, Th2, Th17 cell differentiation under two different conditions, or Treg, TGA beta induced Treg differentiation. So as you can see that under Th1 or Th2 differentiation condition, there was no difference between 37 or 39 degrees incubation or Treg differentiation did not show any difference either. But under two different Th17 cell differentiation, Xiaohu observed that I-17 staining by antibody was nearly double in cells cultured at 39.5 degrees than those cultured at 37 degrees. Then Xiaohu performed RT-PCR to further analyze a bunch of genes associated with Th17 cell differentiation or function. Surprisingly, our RC or our alpha expression was not affected by the increased temperature, but cytokine secretion, in particular IL-17, A and F, IL-22 was all increased, and IL-10 was downregulated. Pathogenic factors, including IL-23 receptor, IL-1 receptors, will all show increased expression. So therefore, fever appear to selectively affect Th17 cell differentiation in vitro and without affecting transcription factor expression, lineage-specific transcription factors, but it has an impact on cytokine and other cell surface receptor, cytokine receptor expression. So this result was reproduced later on by a graduate student in the lab, Lu Ni, and she also reproduced this many times. And Lu then further compared by RNAseq the transcriptome of Th17 cells differentiated under 37 degree or 39.5 degree. She found that as Xiaohu found with RT-PCR analysis, the mRNA expression for I-17A, I-17F, and also other chemokine genes was all increased as a result of culturing at 39.5 degrees. Interesting, if we use this transcriptome analysis to compare with Th17 cells culture in the presence of IL-1, IL-23, and IL-6, the so-called pathogenic in vitro Th17 differentiation condition, Lu found that those cultured at 39.5 degree show more similar gene expression patterns as Th17 cells cultured under pathogenic condition. Furthermore, Lu compared the in vitro culture Th17 cells as those isolated ex vivo. She found that in EAE disease, Th17 cells migrated into CNS show more similarity in the transcriptome as those culture in vitro under 39.5 degrees. But those harvested from non-monopropria of intestine do not show significant similarity in gene transcriptome analysis. So therefore, based on the RNA-seq analysis, Th17 cells induced at a high temperature appear to be similar to pathogenic Th17 cells cultured either in vitro or harvested in vivo, ex vivo. So this suggested that Th17 cells induced at a high temperature may be more pro-inflammatory. To prove this or to examine this, Lu did acute pneumonia lung inflammation model, where she cultured OT2 cells at 37 or 39 degree in vitro, and after differentiation, she transferred these OT2 cells in vivo, followed by intranasal administration of ova protein. So then that will result in acute lung inflammation, mostly dominated by neutrophils. Lu found that OT2 Th17 cells culture under 39.5 degrees show increased ability to induce neutrophil infiltration into bath or into the lung tissue. So indeed, these cells are truly more pro-inflammatory in vivo. So then the question became how T cells sense high temperature and induce more IL-17 and other pro-inflammatory mediators in the expression. So in the literature, it has been shown that simulation is total protein simulation is increased as a result of increased temperature. So Lu confirmed this by using Th17 cells culture under 37 or 39.5 degrees. As you can see, probed by anti-SUMO2 Th17 cells culture at 39.5 degrees indeed show more total SUMO2 conjugation at the proteins, and particularly SMART4 SUMO target also show increased simulation after IP. So then Lu wanted to confirm the functional roles of simulation. She took advantage of UBC9 conditional knockout mice. UBC9 is the only E2 enzyme for simulation. So when you knock out UBC9, you abrogated all the simulation in cells. So then Lu cultured T cell, naive T cells, to differentiate into Th17 cells in vitro. You can see that although white type cells show increased IL-17 expression after culturing at 39.5 degrees, UBC9 knockout T cells did not show any difference. And interestingly, even at 37 degree, UBC9 knockout T cells already show somewhat reduced IL-17 expression, suggesting simulation is important for Th17 cell differentiation. So then simulation is induced or potentiated after culturing T cells at 39.5 degree. What would be the factor that is simulated and also transmitted this signal to induce more IL-17 expression? So I already told you that SMART4 is simulated, and its simulation was increased after T cell culturing at 39.5 degree. Indeed, SMART4 is the target. We first analyzed STAR3 activities and didn't see any difference. And in the TGA beta pathway, SMART4 is an interesting molecule because at 37 degrees, it's selectively required for Treg differentiation, but not for Th17 cell differentiation. So, so this was already reported by our lab in 2008. And Lu reproduced these results. As you can see, at a 37 degree, culturing T cells using different cytokines to potentiate Th17 cell differentiation. Lu found there was no difference between Y type and SMART4 knockout T cells. Surprisingly, at 39.5 degree, although wild-type cells show increased IL-17 secretion, but SMART4 did not show this increase. They show the consistent level of IL-17 expression as they were captured at 37 degree. So therefore, SMART4 may be simulated, and also is required for the increase of IL-17 expression at 39.5 degrees. So then we wanted to examine what residues may be necessary for simulation, or maybe the target of the simulation. This was already reported in the literature. There are there's one lysine, one arginine residues that could be simulated. Lu decided to mutate these two residues to prevent simulation. So then she took wild-type of SMART4 knockout T cells transduced with retrovirus containing either empty vector or wild-type SMART4 or mutant SMART4 that could not be simulated. So she found that at 37 there was no difference, but at 39.5 degree, wild-type SMART4 could rescue the temperature sensitivity in IL-17 expression, although empty vector show this difference. Interestingly, SMART4 mutant did not show any ability to rescue the temperature sensitivity in SMART4 knockout T cells. So therefore, SMART4 simulation may be important for cytokine expression. So, but why is it important? It has been already reported SMART4 simulation regulates its nuclear localization. So, Lu found that when Th17 cells were cultured at 39.5 degree, this high temperature drives almost all of the SMART4 into the nucleus. But when we use sumo mutant SMART4, they lost this ability to respond to 39.5 degree to translocate into the nucleus. So this has put fever to SMART4 simulation and the SMART4 nuclear localization together. And I should mention that these the function of SMART4 was sensitive to TGF beta, because when we block TGF beta using antibody, it abrogates the ability of SMART4 to respond to high temperature. So then, is SMART4 simulation or SMART4 required for Th17 cell development in vivo? So we took advantage that SMART4 was only required for in vitro for cytokine expression following 39.5 degree culture. So Lu cross SMART4 fluoxamides with CD4 to delete SMART4 only specifically in T cells. So in these knockout mice, when you induce EAE, Lu found that they are greatly resistant to EAE disease and also show reduced IL-17 positive cells in the CNS. And interestingly, in the literature, there has been reported that aspirin can reduce EAE score. Lu found that aspirin could also reduce IL-17 positive cells in the CNS of EAE mice. But this effect was not observed in the SMART4 conditional knockout, suggesting that fever that is inhibited by aspirin can be linked with SMART4 in this EAE disease. And we believe that there are many effects of fever in autoimmunity, but it appears that some SMART4 expression in T cells and also its ability to potentiate cytokine expression is important for autoimmunity, at least in the EAE model. And then what is the function or functional roles of SMART4? To address this, Lu did ChIP-seq with SMART4 antibody using Th17 cells cultured at 39.5 degree or 37 degree. So then Lu also combined integrated the analysis with gene that were upregulated in Th17 cells cultured at 39.5 degrees. So then when we combine all these three data sets, we could come up 60 genes that were only bound by SMART4 under high temperature, and whose expression was increased after culturing Th17 cells at 39.5 degree. So there are, within this list, there are I-17A, I-17F, IL-21. These are the genes that are already told you to be affected by high temperature. So it appears that SMART4 could directly regulate cytokine expression in response to high temperature. Interestingly, TGF-beta receptor 1, the upstream molecule, cell surface receptor of SMART4, was also bound by SMART4 and also increased by SMART4, increase at a high temperature. So this suggests that SMART4 not only augments the downstream gene expression, but also may feed forward to enhance its own signaling by increasing TGF-beta receptor expression. So therefore, this work, we believe that has linked one environmental factor, fever, commonly seen in infection and also in autoimmune diseases, with through SMART4 to regulate the pathogenicity of Th17 cells by enhancing the cytokine expression and also the receptor IL-23 and IL-1 expression in T cells to further reinforcing the pro-inflammatory function of Th17 cells, which has implications in autoimmune diseases. Okay, so that's my first part. So the second part, let me touch on the regulation of ILC3. So these are ILC3 belongs to the ILC lineage, and ILC3 is known to express alpha gamma just like Th17 cells.
Chen, I interrupt you 1 sec. There is a little bit of noise, but I think it's the connection. Maybe if you turn off, you just remove the background, the virtual background, or turn off the camera, that the connection will become a little bit better. I mean, we can hear you fine. It's just that maybe we can make it a little bit better.
Okay, is it better? Maybe I can speak more loudly.
Okay. Yeah, we can try that. There was a little bit of
maybe I will use the headset. Maybe I can use headset.
Okay.
Just a second. Hello?
Yes. Hello? Yes.
Okay.
We can hear you there.
Is it better?
Let's try. Yeah, it's better. There's less noise. Yeah.
Okay.
And
all right.
You can do presentation mode.
Yeah. Okay.
For the PowerPoint.
Yeah.
So it's Yeah, perfect. Excellent. Thank you so much. Sorry for the interruption.
It's all right. Thank you. So ILC3, they are known to secrete not only IL-17, but more importantly, IL-22, which will stimulate the epithelial cells to produce antimicrobial peptides, and also is important for the wound healing and repair of the mucosal surface. So the reason we came to this cell type is because we have a long-standing interest in IL-17 family cytokines and the receptors. So in this family, I should say that describe that these cytokines serve at the interface of lymphocytes and the epithelium. For example, IL-17 and IL-17F, I already told you, are produced by lymphocytes. They bind to cell surface receptors expressed by epithelial cells to induce inflammation and also gene expression. On the other hand, IL-17B, C, and E are known to be expressed by epithelial cells. Not only they act to signal within the epithelium, but also they also signal and regulate particular type of lymphocytes. For example, IL-17E, also known as IL-25, its a receptor is selectively expressed by type 2 lymphocytes. On the other hand, IL-17C binds to IL-17RE, which is highly expressed by type 3 lymphocytes, including gamma delta 17 and Th17 cells. So these cytokines have emerged as critical players in mucosal diseases, and also they are targeted by various antibody drugs, and that have been already approved in the clinic or are being tested in the clinic. So, so in this family, there's often cytokine called IL-17D. This cytokine, of course, share homology with the other IL-17 cytokine family, but its receptor and function was not clear. Which is the subject I will discuss right now. So former postdoc in the lab, Yang Hee Lee, coming from Seoul, South Korea, she decided to work on IL-17D, and she took IL-17D knockout mice and treated these mice with DSS. And for 5 days, after 5 days, she stopped and changed to normal water and as drinking water. You can see that white mice, after 5 days, they started to regain weight, and the epithelium started to be repaired because DSS will kill the epithelium cells. On the other hand, IL-17D knockout mice show continuous weight loss, and these in the intestine, these mice show worse inflammation and also impaired wound repair. Then Heian left the lab. This project was taken over by another postdoc, Jinling Huang, in the lab. Jinling not only reproduced the DSS phenotypes, but also she examined the expression of IL-17D in different tissues. You can see that IL-17D not only mRNA not only was expressed in spleen, but also highly expressed in lung and the intestine tissues. So then Jinling further examined the expression of IL-17D in epithelial cells and also in CD45 positive leukocytes in the intestine. She found that IL-17D was mostly expressed by CD45 negative epithelium cells. And interestingly, in the expression of I-17D mRNA, there was no difference between SPF or germ-free mice, suggesting that this expression was not regulated by microbiota. So to prove that I-17D expression by epithelial cells may be most important for its function, Jilin did bone marrow transfer experiments where she took Y-type or knockout I-17D knockout bone marrow cells and transferred into irradiated Y-type or I-17D knockout mice. And then she further induced DSS colitis, acute colitis, by feeding DSS. So as summarized here, using colonans as an example of inflammation in the intestine, you can appreciate that no matter what the donor cells were, as long as the recipient mice were knockout, they all show the worsened DSS colitis disease. So this proved that a non-hemopoietic source of I-17D may be important for its function to regulate wound repair following DSS treatment. So then at that point, we don't know the target of all the receptor for I-17D. Jilin has at that time extensively analyzed immune cells in the intestine. She didn't find any difference in the numbers of cytokine expression by CD4, CD8, or gamma delta T cells. And so then we decided to focus on ILC3 because as you can see here, ILC3 numbers were not affected in the I-17D knockout mice. But on the other hand, the cytokine expression, IL-22 expression, was reduced. In the intestine, ILC3 can be divided into three major subsets, those expressing CD4 or NKp46 or without both expression. So in these three subsets, Jilin found that they all show reduced IL-22 expression. So suggesting that I-17D may not be necessary for ILC3 development or expansion, but is required for the ability to secrete IL-22. So then Jilin took the total ILC from Y-type or knockout intestine and did RNA-seq analysis. She found, again, reduced IL-22 expression, and also interestingly, HR expression was reduced, and also the expression of a surface receptor called CD93 was also reduced in IC3 cells derived from I-17D knockout mice. So then we thought I-17C could perhaps serve as the target of I-17D, but is this a direct target? Before we investigated that, Jilin applied two other models to I-17D knockout mice. First is the colon cancer model induced by DSS-LOM. You can see here that I-17D knockout mice show more tumors as well as increased sizes of tumors. So in the tumor model, Jilin found that I-17D knockout mice show reduced ILC3 cell numbers, although they do not differ from Y-type in their Th17 cell numbers. So the second model Jilin applied was the Citrobacter rotation infection. White type mice show constant weight, but the I-17D knockout mice show reduced weight after the infection, and also they show 1 log higher bacteria count in the feces, in the fecal. And also the I-17D knockout mice show greater reduced IL-22 expression after Citrobacter rotation infection. So in these 2 models, we all found abnormalities associated with I-17D deficiency. On the other hand, both models also show consistent ILC3 phenotypes, suggesting that I-17D may perhaps regulate ILC3, either in the numbers or in their function. So to prove that I-17D may directly regulate ILC3, Jilin prepared an I-17D recombinant protein by fusing I-17D with human IgG Fc tag. So you could see that these I-17D recombinant protein did not bind to T cells from intestine, but it binds to a significant portion of ILC3 cells from the intestine. These suggest that I-17D may directly bind to ILC3 and may also regulate ILC3. To further study this, we have to identify the receptor. Jilin took a heroic effort. She screened all the cell lines in the lab and found a macrophage cell line that could be bound by I-17D. So then she, after the binding of I-17D recombinant protein to this cell line, she cross-linked these recombinant protein. So you can see the I-17D recombinant proteins and also a larger size of protein complex cross-link. And Jilin subject this band to mass spec analysis and then identify all the transmembrane proteins that were obtained from the mass spec analysis and cloned the cDNA and tested the binding of I-17D to transfectants with this cDNA. So after this, it was obvious that I-17D could bind to CD93. So you could see that when Jilin expressed I-17RA, it could be bound by I-17A, but not by I-17D. On the other hand, CD93 transfectant could not be bound by I-17A, but it could be bound by I-17D significantly. So also Jilin proved that in solution, I-17D and CD93 recombinant proteins can directly interact. So therefore, I-17D may bind to CD93 that is produced by ILC3 in our RNAseq analysis. And Jilin further analyzed CD93 expression in the lymphocytes from the gut. She found that only ILC3 express a significant degree of CD93. Interestingly, the cells expressing CD93 correlated with IL-22 expression. And when Jilin isolated cells and performed RNA-seq, she found that these cells are functionally more mature than those which did not express CD93. So therefore, CD93 may be a maturation marker on ILC3. So Jilin also created CD93 knockout. As a result of the knockout in the intestinal ILC3, you don't see antibody binding by CD93. On the other hand, I-17D binding to ILC3 was also lost in CD93 knockout ILC3. And in these mice, gene-induced DSS colitis and CD93 germline knockout show greater weight loss and reduced colon length, and consistent with I-17D knockout mice. Furthermore, to prove that CD93 expression in ILC3 is most important target mediated by regulated by I-17D. Gene created also CD93 flux mice and cross these mice with RRC Cree to disrupt CD93 gene, specifically in O type 3 lymphocytes. In the intestine, it's obvious that the conditional knockout mice already show reduced IL-22 expression, and following DSS colitis induction, they show increased weight loss and also reduced IL-22 expression. So to summarize this part, we believe that we identify the function and the receptor for I-17D. I-17D is constitutively produced by intestinal epithelial cells. This expression was independent of microbiota. I-17D selectively binds to ILC3 because ILC3 express its receptor CD93. CD93 does not belong to the I-17R family. So this is a very unique regulation. So we know that ILC3 arise from bone marrow precursors, and when these precursors come to tissues, they need to functionally mature. So we believe that I-17D binding to CD93 will further mature ILC3 to equip them with the capacity to secrete IL-22 upon environmental stimulation. IL-22 secretion was important for intestine epithelial cells to produce antimicrobial peptides to repair the epithelium following DSS colitis, and also to control inflammation associated colon cancer. So therefore, not only we identify the receptor for I-17D, but we show that it is again, it serves I-17D and CD93 axis serves as the interface between epithelium and intestinal lymphocytes to crosstalk and to regulate each other in homeostasis and also in diseases. To end, let me thank the postdoc and students associated with the work. The first part on fever was initiated by our associate professor Xiaohu Wang in the lab, and then continued by Lu Ni, former graduate student in the lab. The second part on I-17D was mostly done by Jilin Huang, a postdoc in the lab. Both Lu Ni and Jilin got help from Xiaohong Zhao, a bioinformatic analyst in our lab. We had help from Xinquan Wang's lab at the Tsinghua University School of Life Sciences, where his postdoc Jiwan Ge helped Jinlin to demonstrate the direct binding of I-17D and CD93 in solution. So let me end by thanking Global IMMUNOTALK for the invitation, and also to the audience for listening. Thank you.
Chen, thank you so much. That was a terrific talk, and thank you for sharing these two amazing stories, both the one with the fever and the IL-17D. These are great advances, and I'm sure people will have questions for you. And as always, and to make this forum as egalitarian as possible, so we can have people from different time zones also ask questions. We do the questions via Twitter, and so you should see now in the in Twitter, if you search for Global IMMUNOTALKs account, a tweet that says "Ask questions for Dr. Chen Dong here." So to ask a question, you just reply to that tweet with your question and mention the hashtag Global Immuno. And then Chen has generously agreed to answer the questions, and the account that will be used is the IMMUNOSpeaker account. So Chen, thank you so much again. It was a pleasure and an honor to have you in the list of Global IMMUNOSpeakers. So thank you.
Thank you.
Okay.
Thank you.
Okay.
Thank you.
Take care, everyone, and I'll see you.
Take care. Stay healthy.
Bye.
Bye.